Chapter 8, Techniques of Recombinant DNA Technology. The learning objectives for this topic are: describe the purpose and application of the polymerase chain reaction, explain how researchers use DNA probes to identify recombinant cells, describe the process and use of gel electrophoresis, particularly as it is used in a southern blot, identify the manufacture and use of DNA microarrays, and list and explain 3 artificial techniques for introducing DNA into cells. So, in this topic, we're going to go over PCR, or polymerase chain reaction, selecting a clone of recombinant cells, gel electrophoresis and southern blot, DNA microarrays, and inserting DNA into cells. So, for PCR, basically what we're doing is we're making a photocopier for DNA. So here's how it works. First off, remember that enzymes work outside of cells as well as inside of cells, and we make use of this in PCR, or polymerase chain reaction. So here's the gene of interest, and it's in a double-stranded DNA. So we heat it to 94 degrees Celsius. That's enough to break the hydrogen bonds, but not hot enough to break the sugar phosphate bonds. We don't want to do that. We call this denaturation. So then we add deoxyribonucleotide triphosphates. Remember, during replication you got to have triphosphates to provide the energy for the polymerization. We put in the primer. The primer is the beginning of the gene of interest. So remember that DNA polymerase has got to have a primer. So we put a DNA primer in instead of an RNA primer. And we put in DNA polymerase that can withstand the high heats. So we take them from hypothermophiles. So the DNA polymerase goes in, polymerizes complementary strands to go with the denatured strands of DNA. So then, using the PCR, for each cycle, we double our strands. So in the first cycle, we go from 1 strand of double-stranded DNA to 2 double strands. Second cycle, we go to 4 double strands, 8 double strands. So basically, we're doing exponential expansion, just as if the cells were dividing. And we can make all sorts of copies of the gene of interest so that it's easier to study. It's a whole lot easier to study a whole bunch of a gene as opposed to just one copy that you've got to find somewhere in your container. Next up, selecting a clone of recombinant cells. This is when you're wanting to make sure that you're studying the actual gene that you're interested in. So you got to search the library. And we do that by using a labeled probe. Now remember, back in the previous topic we talked about how we could make probes using synthetic nucleic acids. Basically, we set up a stretch of single-stranded DNA that's exactly what we're looking for. Actually, it's complementary to what we're looking for. And we label it. We can label it with green fluorescent protein. We can label it with radionucleotides. Then we can go into our gene library and see where the probe sticks. So wherever we see the label, whether it's radioactivity or whether it's a green fluorescent protein, then we know that's the gene that we're looking for, and we don't have to mess with the other cells, the other clones in our library. Let's talk about gel electrophoresis. This is how we separate different sized chunks of DNA. So we've run our sample through PCR. We've got several different sizes of fragments. What we do is we put it in agarose gel. Agarose gel is like agar gel, but it's a little more floppy. And we cut little holes or wells. They're actually wells because they're not holes. They don't go all the way to the bottom. We don't want our DNA to fall out. And we put our sample into these wells. Then we apply electricity across the gel. So we have our negative end here, we have our positive end here. What's the charge on the DNA? That's right, it's negative, so it will move toward the positive pole. So we always include a control, and we call this a ladder, where we have known sized fragments of DNA. And you'll notice that the bigger ones are staying closer to the negative side. Does that mean that they're more positive? No, it means they're bigger, and they're having harder time wiggling through the gel. Think of it this way. The gel is all full of little itty-bitty microscopic holes. What's going to move through it faster, a big chunk of DNA or a little chunk of DNA? Well, a little chunk of DNA is going to move through. Now, once we've separated out our DNA fragments and we don't want to run them off the end of our gel and into our solution, we can take the gel and apply a DNA stain, which will fluoresce under UV light, and then we can study the patterns that are made. Now, in 201, remember we did some forensic research and all sorts of fun stuff with the gel. That's what we use the gels for. But there are times when we want something a little more stable than the agarose gel, which is kind of like thick jello. So what we do is a southern blot. A southern blot is, we basically do an electrophoresis, and then we transfer the DNA fragments to nitrocellulose membrane, which is kind of like photographic paper, and it's a whole lot easier to handle. So let's go through a hypothetical situation in which we would do a southern blot. Suppose I'm wanting to take a look at clone number 1 to see if it has the gene that I want to study. So I take my DNA molecules from clone number 1, and I've isolated the DNA and I expose them to restriction enzymes. So they're going to cut where they were when I initially inserted the DNA, the gene fragments. So then I put them on my gel and I run my electrophoresis and I separate out the fragments. Then I take my gel, put it on top of the nitrocellulose and put it on top of absorbent material, and the wicking action will draw the DNA into the nitrocellulose, which is a whole lot easier to use. So then I can take the nitrocellulose membrane and I can add radioactive probes that are looking for the gene of interest. Then I can take the nitrocellulose and I can incubate it with film. It'll show up which bands show the gene that I'm actually looking for. And in this case, oh, clone number 1 has the gene I'm looking for. I don't have to check the others. If it didn't show up, then I would run the same process with clone number 2 and so on until I find the gene I'm looking for. There are variations on the southern blot. There are other things that you can run on gels other than just DNA. RNA leaps to mind. RNA acts just like DNA on an agarose gel, and you can transfer them to nitrocellulose and do exactly the same thing. Well, the fellow who came up with the southern blot, in which you transfer DNA from an agarose gel to nitrocellulose, his last name was Southern. So, of course, what do we name the same procedure for RNA? The northern blot. Yes, scientists have senses of humor too. You can also do the same thing with protein, and that would be a western blot. Get it? Out west we have lots of cattle, lots of protein. Yeah, whatever. On to DNA microarrays. Basically, what we do is, somebody got tired of using one probe on a southern blot. So what they came up with is, let's do all sorts of sequences of probes. Basically, we're going to make a whole bunch of probes, and we're going to put them on a glass slide or on a computer chip. That's how they did it the first time. And they take these probes and they attach them in known spots. Then they take the DNA from a sample, and we've fluorescently labeled them so that the more DNA that grabs a hold of the probe, the more it moves from either black to red, which means only a few probes grabbed a hold, to green, which means a whole bunch of them grabbed. And you can get a lot of information all at once. And you can run samples that aren't pure. So, say for example, I take a sample of a soil sample, and I take probes from known genera of bacteria. And I take those probes and stick them all over my little slide there. And then I chop up my DNA samples from my soil sample. Then I do PCR with my soil sample DNA using fluorescently labeled nucleotides. So then I run them past my probes and I get a pattern like this, and I can look at my entire soil sample all at one time. Saves time. Now, because the pattern is so complex, because we can put thousands of probes on, we generally use a computer to analyze the data for us. Now on to inserting DNA into cells. So I've got a plasmid, it's got a gene that I know that I want to study, and now I want to put it into a cell. So the first thing that we can do is called electroporation. This is what we would do to cells that are not naturally competent. They don't have the transporter protein to bring in big chunks of DNA. Now, with organisms that have a thick cell wall, such as algae and fungi, we got to remove the thick cell walls, and we call that a protoplast. So we put the DNA in the environment, and we run an electrical current through the media, and that punches holes in the cell membrane. Well, the holes are repaired over time, and the DNA hopefully has moved into the cells. Now we can take and do protoplast fusion instead. So say we only got one or two cells that actually got the DNA that we're interested in. Well, we can take and remove the cell walls from other cells, and when they bump into each other, their membranes fuse. So we're able to move the gene from one cell to another cell by having them fuse. And they do a lot of that in plant research. And polyethylene glycol increases the rate of fusion. You may have heard of polyethylene glycol. It's used in lots of other functions. Injection. Injection is an interesting one. Here's what I've mentioned before. Here is a target cell. Here's the nucleus. This micrograph is phase contrast. So we take a small little pipette, apply a gentle suction to hold the cell in place, and then we come in with a micropipette to go in and inject the DNA directly into the target cell's nucleus. Now this other one, I have no idea how this works, because it just makes me laugh just to think of it. They have a gun, and they get an actual 22 caliber shell, and they have a nylon projectile, and then they have DNA coated beads. So they fire off the bullet, slams the nylon projectile into the end, and it causes the beads to come flying out, and basically you shoot your bacterium. And can I just say, this is a huge bacterium if that's a 22 caliber shell. I'm sure they blew this up just so you could see the DNA coated beads. Well, that's it for this topic. Here is a reminder of the learning objectives for this topic. Make sure you write down any questions you may have.